Yeah, so today I want to be talking about webKnossos, which is an open source tool for collaborative image analysis on very large three-dimensional data sets. So I'm Norman, I'm the co-founder of Scalable Minds. You can find me on Twitter at NormanRZ. And Scalable Minds builds image analysis tool for neuroscientists. One of them is webKnossos, which is an open source tool that I want to talk about today. And the other one is Voxelytics, which is an automated toolbox for reconstruction. It can do stitching, alignment of 3D data sets, and of course, also automated segmentation of neurons, synapse detection, organelles, and everything like that. We collaborate with a bunch of labs from all over the world. Two of them are the Helmstetter group at the Max Planck Institute for Brain Research in Frankfurt, Germany. And the other one is Andrea Schäfer lab at the Crick Institute. And in my talk, I will be showing some of the data that they've acquired and worked with webKnossos. So let's dive into webKnossos. I first want to talk about the data sharing and manual annotation features of webKnossos. So as the name suggests, it's a web-based tool. So you can open up a browser and easily browse through very large data sets from moderate internet connection. Like the animation here is not sped up. This is a 50 terabyte data set that you might have seen. It has been published 3 years ago. And it's around 50 terabytes in size. But webKnossos can also handle larger data sets closer to a petabyte or even larger. And the great thing about the web technology is that you can send links to collaborators, and these collaborators can then help you with annotation work. webKnossos supports basic brush and lasso tools for creating volume annotations, as you can see here. And it also has support for larger annotation crowds with a task management system, so you can distribute tasks over a crowd of annotators and collect all of the results back in and work from there. And you can also have redundancy features in there so that you can do quality control on top of that. webKnossos also supports skeleton annotations, which is quite popular for tracing neurites. So skeletons are these line segment representations of a graph structure, and webKnossos has a fast mode of creating those, and this is what we call the flight mode. In the flight mode, the data is projected onto a sphere, which makes it easier to rotate around and navigate as you're flying through the data. And in our Nature Methods publication a couple of years ago, we measured that trained annotators can get very fast at doing that, both for axons and also for dendrites. And with all of that, you can collaborate on very large projects. This is a manual reconstructed project you can see here. And because of all of the crowdsourcing features, you can get quite large projects done. But if you want to go to the very large projects, then you need some machine learning workflows. And webKnossos doesn't have machine learning features built right into it, but it can greatly assist the workflow in working with these large scale reconstruction projects. The first way it can do that is maybe obvious through generating training data. Again, you can use the task management system to distribute work, collect it back together, and then train a model in your favorite deep learning toolbox. Here you can see a nuclei prediction that we've done with Voxelytics. And you can see that webKnossos has support for overlaying multiple layers. So in the background, we see the raw EM data. And on top of that, you can see the prediction layer of our nuclei prediction in blue. And we can use the histogram sliders here to find a good threshold. And we could use this threshold then to make a segmentation out of these probability maps. Here's another example. This time we have mitochondria in red and synapses in blue. Again, automatically reconstructed. And again, we're using the histogram sliders to adjust the threshold. And again, the great thing is that this comes with a URL. So you can send this exact scene at the exact location to your collaborator. And then you can together look at the data and work on any issues that might still be in the data. webKnossos also works great with instance segmentations. Here you can see a reconstruction of neurons. And in the first part, we saw an over-segmentation. Many segmentation pipelines work by first having an over-segmentation where the objects of interest are actually split, and then have a separate automated step called agglomeration that piece these supervoxels back together. And webKnossos has support for visualizing these supervoxels. Like we can select a mapping here, and then we will see the agglomeration applied. And this is great for debugging again, because you can easily switch between one mapping to the next and find the one that works best for your data. You can also proofread segmentations with webKnossos. Here we can see we're using the skeleton annotation tools to fix an over-segmentation, so we call the merger mode. And if you already have a representation of a supervoxel graph, you can also load that into webKnossos and edit it from there. And of course, you can also use the brush tools again to refine any contours that may be off. And once you have all that in place and you're happy with the segmentation, you can use webKnossos to visualize meshes. These meshes can be either pre-computed by an external tool, or they can be computed from webKnossos directly on demand. And you can visualize them. You can take pictures directly from webKnossos, but you can also download them and load them into your favorite visualization tool like Mira or Blender and make pictures from there for your publication. And once your publication is ready, you can also share your data alongside your publication. You can put links into your paper to specific locations in webKnossos, maybe from a figure caption, and then your readers will be able to explore the data and the story that you're trying to tell interactively from webKnossos and directly by just clicking on the link. And we do have a gallery of published data sets on webknossos.org, and we're always looking for more data sets to add to that list. And with that, I would like to thank you for your attention. Again, there is on webknossos.org, you can try out webKnossos for yourself. There are a bunch of published data sets that you can try, but you can also create an account and upload your own data set. We do have a pilot program where you can sign up to, and you will get up to 10 terabytes of free storage to work with your data there. And my company also offers services for manual annotations and automated alignment and segmentation for three-dimensional data sets. Please do let me know if that's of interest to you. And you can contact me on Twitter, via email, or at the symposium. Thanks.
Thank you very much, Norman. It's really wonderful tool. So we have some data hosted there, and it's really nice to explore it. If I understand correctly, people can also generate ground truth for their, like if they want to do automatic segmentation. Because I know a few people that, that they try to make their own tools, and that could be also very, very useful. I don't see any questions in the Q&A box. If someone has a question now something is popping. Sabrina is asking if you have plans to extend the software to cryotomograms. I guess it's already there.
Yeah, absolutely. Yeah. So I mean, it works with 3D data sets of all kinds. It works well for like serial block phase or FIPSEM. We also have like X-ray data in there. We also can support fluorescence data sets with multiple channels, and you can overlay them. You can also overlay fluorescence layers on top of EM, and so it's quite versatile in what kind of data sets it can handle.
Maybe I wonder if Sabrina's thinking about cryo-EM, like for structures. I'm not sure, but
yeah, I mean, it's certainly something we can look into.
You can chat in the speaker session.
Yeah.
Okay, thanks so much. So now